Okay, well that beginning stuff doesn't need to be recorded now. We are recording. Okay, great. All right, so good morning, everybody. We are going to continue our conversation about cells. This is actually one of my favorite units because I think it's a little more dynamic. It kind of tells a story as we go through it. So I think it has some nice pieces that have continuity with them. So we had... I feel like I have to sneeze and it's not going to give it to me, which is very unsatisfying. Yep. My mask kind of pinches my nose, so it's just not going to let it happen. Okay, so we had just finished up some of the main components of cell theory. So we talked about what is it? Man, come on. How cells are the base unit of being living things. We talked about how big and small can cells be. And so now, right, we're back to our roadmap. We want to focus on what are the parts that all cells have. Okay, and again, we've talked about this a little bit before. Okay, so we have some base footing here. Okay, and so basically we're going to take the things that we know and we're going to add to that or expand upon that. Okay, so here's the thing we know. Okay, we know that all cells have a plasma membrane. Okay, now we keep talking about this being sort of the grocery bag around the cell. Okay, so we knew that from our very first unit ever. Okay, so this is sort of that protective layer around the cell that keeps all the contents inside. Okay, now we added to that in our biomolecules unit, right? We knew then that this plasma membrane was made out of a phospholipid. Okay, very specifically, we have I'm drawing, don't be rude. We had our phospholipid head, right? Our fatty acid tails. Our fatty acid tails, remember, are non-polar. Right? Non-polar things don't play well with others. So they're afraid. So they tuck themselves into the closet together. Creating that safe wall space, okay, through which very few things can pass. And then we have the other phosphate head. All right. So we have the two polar pieces on the outside. Okay, so this is something we learned in biomolecules. So this is something we added. So both of these pieces are familiar. So the only new thing here is part B. Okay, and that's this idea that the membrane itself is something we call selectively permeable. Okay, permeable refers to how well something can pass through. Okay, so we generally talked about how because of this nonpolar center, right, we don't like things to pass through. Okay, in fact, we'll talk about in our next mini unit that pretty much not very much can get through here at all. Okay, because they don't play well with others. Okay, so by selectively permeable, we basically mean it's an exclusive club. So this membrane is going to pick and choose the kinds of things that can get through it. Okay, very few things will get through the membrane itself, and everything else is going to have to use a gate. aka a protein. Okay, and remember that's the whole idea of having these membranes, right? We want to keep the stuff inside the cell safe. Okay, so we don't want to just let the everyday rabble in here. Okay, we want to be able to keep out bacteria and viruses and nasty abrasive things. Okay, but we do need to let in things like food. Okay, so we're selectively permeable. We're going to allow in good things. Air and food, keep out bad things, disease. Okay, that feel pretty good? Okay. Other stuff all cells have, cytoplasm. Okay, so we've mentioned this in the past, okay, sort of in passing. So when we talked about our biomolecules, we talked about cytoplasm as that wet stuff that was inside the cell. And so this is why it was okay to have those polar heads facing the inside the cell, because there was all that wet stuff in there. This is that wet stuff. Okay. But more importantly, or more specifically, the cytoplasm sort of ends up becoming like this gelatinous ocean that we'll look at, on which gesticulating a lot today, on which everything else, okay, all of our organelles, everything that's inside the cell kind of floats. Sort of like how we imagine a real ocean. You have all the boats and the buoys and the fish kind of floating out there, living their best life. Okay? This is a lot how the cytoplasm works too. Okay? Everything in a cell floats out there, living suspended. Okay, it's a little thicker than plain water, but it's serving the same function. Okay. So we're going to see this prefix a lot, cyto, okay, literally means cell. So it kind of translates to cell material, cell goo, quite literally. Don't let Dr. Watson know I called it that. All right, next on the list, and we've talked about these before too, organelles. Okay, remember that organelles literally mean many organs. And that's how they function. If we think about a living cell, right? A single cell can be a living thing, okay? On that scale, then, anything inside it has to be working to function to keep it alive, just like the organs in your body function to keep you alive. So each of the organelles inside the cell performs some kind of specialized function. And we all know at least one, right? The mitochondria, the powerhouse of the cell. Okay? It's possible to forget it. And we'll talk more in detail about what exactly that means. But there are a lot of them that do those sorts of things. So we're going to go through at the end of this and look at a few select ones that are interesting and have very specific jobs. But for right now, just know that there are several, okay, that function like mini organs and that do jobs to keep the cell alive and working. Everybody still feel good? Okay. Something else we're familiar with? DNA. Okay? This is also right off of our biomolecules topic, right? So DNA was one of the 2 forms of nucleic acids that we had, okay? Specifically the one that deals with information storage. Okay, so we're going to see here in a minute that the way DNA looks might be a little different. Okay, the way it's housed might be a little different, but it always has to be there. Okay, there has to be a version of this in which, for all cells, we can keep all of the information for how to be a cell. Okay, does that make sense? Right? You have to be able to keep all of the books on how to not die and be able to pass that on. Okay, so far, okay, we've added a few details. We haven't smacked our faces into too much that's brand new. Okay, any questions about any of that business? Okay, so remember at the very beginning in our intro slide, I mentioned that there were 2 major different types of cells. Okay, and so this is where we want to talk about this because, as I mentioned, even I, for once, am going to have a little bit of mammal bias for the most of this. Okay, so I want to make sure that we do stop and recognize that there are 2 types of cells and that they do look different. Okay. All right, so our 2 different types of cells are prokaryotes. I'm drawing. So rude. Prokaryotes. Okay. Think pro, kind of like prototype. Okay, meaning like early. Okay, so our early cells. Not always entirely true, but that's where the word comes from. Okay. These are going to be our small and simpler cells, like our single-celled organisms. As we'll find. And then our eukaryotes. Okay. This is again where the mammal bias comes through because we named these things, right? Okay. So the prefix "eu" means true. Sounds pretty biased, right? Okay. They're both cells. They're both real cells. But eukaryotes are the true cells. Okay. So as you might imagine, these are the kinds of cells that we are. Okay. So these tend to be larger and more complex. And these are the ones that, for the most part, we're going to spend most of the time talking about because they are more complex and, of course, they're ours, right? So we want to know a little bit more about them. Okay, so let's look at some of the details of these cells and how we can tell them apart. Okay, so we're going to start by looking at prokaryotes first. All right, so a prokaryotic cell. So as I mentioned, these guys tend to be smaller. Okay, and again, most of these tend to be our single-cell organisms, right? So these are our bacteria and our protists for the most part. Okay. Now, aside from being small, there are a couple other key features that you're going to use to be able to recognize these as prokaryotic cells. Okay, the first of which is these guys have a cell wall. Now, there are eukaryotic cells that also have cell walls, specifically like plant cells, but this cell wall is different in that it's covered in a sticky capsule. Okay, think very Dr. Mario kind of business. Okay, so this sticky capsule allows them to maneuver through their environment, okay, and protects them. okay, from the world around them. So if we imagine those little guys like swimming in the lake, okay, having that sticky capsule on them keeps other organisms from being able to eat them, or be less likely for them to be able to eat them, because who wants to eat something that's sticky, right? So it makes them less likely to be prey items. Okay, whereas a plant cell isn't exactly free floating, right, in the water, so it doesn't need to be sticky. Okay, our DNA, now we still have DNA. Remember, that was one of our core pieces that you have to have in order to be a cell. So we have DNA, but we don't have a nucleus. Okay, this stuff is free floating. It's very 1970s hippie. Okay, it's just wherever, man. Okay, so I'll show you some pictures of this when we look at these both, but when we look at the DNA, it is very literally just kind of rocking it out and loose and in the open. Okay, whereas when we find and compare this to a eukaryotic cell, those of us that have seen these before, okay, most eukaryotic cells have a nucleus. It's very protected and centralized and compact, okay, in its own mini little house. Okay. In fact, as we'll find here, structure is not something prokaryotes do. Okay. Okay, so even their organelles do not have membranes. Okay, so they have ultimately a little bit less diversity in the overall types of organelles they have, and they're not membrane bound. Okay, so the whole inside of these guys are a lot more loosey-goosey. They go where the wind takes them. Okay, so the only thing that's really structural about these guys is their cell wall, which is rigid and sticky. That feel okay, generally? Okay. Let's compare this to our eukaryotic cells, right? A good comparison always helps stuff make a lot of sense. No. This. Okay. Now, our eukaryotic cells tend to be larger. That's easy enough. Okay. The big keys here, though, is our cell walls are not always present. Okay, so things like fungi, not always. Animal cells do not, right? So your cells do not have a cell wall at all, right? All we have, and all of these have the plasma membrane, right? Because that was one of the key features. So we all have a plasma membrane, okay? It's just whether or not there's the addition of an extra protective wall layer, okay? So our cells don't have anything extra. Plant cells will have an extra layer. It's basically like a Lego block. Just allows them to stack up so you can have like a straight stem. But again, if we think about a plant cell, okay, they're not going to be sticky and nasty, okay, because they're not free floating in the water. Okay, so they don't have to worry about predators. That's why the whole plant has things like thorns or something on it. They have different defenses. So even if they do have cell walls for things like structure, they're not going to have these sticky capsules on them. Okay, or the cell wall may be absent completely, like our cells do. Just a plasma membrane. Okay. Once we get inside our cells, there is a ton of structure always. Okay? So the DNA is inside a nucleus. And we'll talk a lot about the nucleus. But basically, if we think about that DNA being the super books all stacked together to contain the information, we can think of the nucleus being the walls of the library. Okay? Thick and protective, making sure that nothing ever happens to that information. Keeps it safe and secure and in one specific spot in the cell. Okay? So not free-floating like it is in our prokaryotes. Okay, it's all tight and contained. Okay, our eukaryotic cells have some non-membrane bound organism or organelles. Okay, so for example, they both have ribosomes, okay, which have no membranes, but we have membrane-bound organelles. And we have a lot of structures. So we have things like the Golgi body and mitochondria. Okay, all of these things that have thick membranes that allow things to pass across them.
So online, somebody said that she would assume that the organelles in eukaryotic cells have a specific place, so they just don't float in the cytoplasm. Would that be correct?
This is correct, and we'll talk about that as well. But so for eukaryotic cells, we have cytoskeleton, and so that's one of the last slides we'll look at, but specifically anchors in roadways that hold the organelles in place. So you're absolutely correct. There's a lot of structure when it comes to eukaryotic cells. And there's some, but very little when it comes to prokaryotic. Okay, so here's a prokaryotic cell. So we can see on the outside, we have that cell wall, and it's very, very thick. We can see that sort of capsule shape. It looks almost a bit like a medicine capsule, kind of what you would picture. Okay, and it has sticky bits on it. Okay, I know I wouldn't want to put this in my mouth. Okay, now on the inside, it does look a bit like Hot Mess Express. Okay, so we do have, okay, this purple bit is the DNA. It's there. Okay, it's all together, so it's organized in a fashion. Okay, but it's not exactly all in one place per se. Okay, it's not in a house or anything. It's just kind of like there. Okay, we're hanging out in a group, but we're taking up 90% of the whole thing, right? Okay, and we have some organelles. My arrow's doing a bad job. I'm trying to point to the dots. Okay, so those are ribosomes, I suspect. Yes. Okay. Which, as we'll find, are really important organelles. Those make proteins. Okay, but those are organelles, as you can tell, they're not very big. Okay. They don't have thick membranes on them. They do important jobs. Okay, but there's not a ton of structure. Okay, everyone's just living its best life. Okay, there's enough there for the cell to survive, right? But if we compare that to a eukaryotic cell, we get a much different story. Okay, so still in purple here, we have our DNA, okay, the thready part in this case. We can focus on that, the actual DNA. Okay, well, that's encased in a house. Okay, our nucleus, which is keeping it safe and centralized in one tight, specific location. Okay? As the person online indicated, all of our organelles are also in very specific locations. Okay? Many of which have these very clear, if we focus on, for example, our endoplasmic reticulum and our Golgi body, very clear membranes on them. Okay? We still have ribosomes. We can see the little dots here. So those still exist. We still have unbound organelles. We still have to make proteins, so we still have to have those. Okay, but we also have obvious membranes here too. There's a lot more organization and there's a lot more structure. And we are a lot bigger. And of course, in this case, this is an animal cell, so we have no cell wall at all. But in either case, we see the absence of that sticky, ferocious-looking capsule. Okay. The difference between these two feel pretty good? Okay, any questions? And very good observation online. Okay. I was not prepared for that. We haven't done one of these for a while. This will be good. All right, so grab out your, whatever it is you use to do your mentees. I used to be able to do these right in the PowerPoint, which is why that does that. Okay, so I know we haven't done these for a while, so this will be nice. So remember, we're going to go to menti.com. Yellow does not work when you're on white. Menti.com. Okay, and it's going to ask you for a code. So here's your code, 2617050. Okay, now without looking at your notes, okay, you're going to tell me which is the best answer for not a feature. Okay, so which thing is least likely to be indicative of a eukaryotic cell? Okay. Yeah, pink's fine. All right, so let's take a peek at this. So why didn't we pick things like nucleus? Why didn't we like nucleus? So all our euks have this? Oh yeah, so this is how we organize our DNA, right? Okay, in fact, this is a twofold thing that we'll know is there. One, this is a special thing for DNA. And two, this also has membranes. Which is also a pretty eukaryotic specific thing. What about the cell membrane? We didn't like that one either. Why didn't we pick that? All cells have to have this, right? So it definitely can't be that for either of them. Okay, so that leaves us with our two here. Okay, so let's look at cell wall next. This is our second least favorite, or least favorite, whatever. Why was this your next least popular one? For those of you that chose the yellow box, why didn't you choose cell wall? Some eukaryotes can have a cell wall. How does this cell wall differ, though? Not sticky. Right, so that's really key here is we want to look at how this is worded. Okay, so this is our best answer, is most of the organelles should have a membrane, right? All right, so you guys did a good job with this. Get out, I can get out. How do I get back? This side. All right. Okay, so any questions about any of that before we move forward? Okay, so the last thing we're going to do is go through, so like I said, I'm going to use a little mammal bias here. Listen, don't do that. Don't. And focus on that eukaryotic cell, okay? So that's the kind of cells we have. Okay, they're also the more complex ones. There's more to talk about. They're also the kinds of cells we're going to be focused on for the rest of this semester when we talk about mitosis and DNA and all of that business. So what we're going to do is take a tour of this cell. We're really going to focus on all the different types of organelles and the functions that these organelles have. And so we're going to start from the inside, okay, really focusing on the nucleus and work our way out, okay? And for the most part, we're going to make the creation journey, okay? And then we're going to add in some other organelles so you can see what a lot of these major organelles do, okay? So what you want to do, remember, I'm always going to try to give us some hints on how we can focus on this, is work on knowing this journey. Think of it as like a road trip, okay? So could you trace a path from a start to the finish if we were going to make something or if we were going to tear it apart? Okay? And that's a really good way to think about being able to name these things. And it kind of gets you away from just having to memorize the Golgi body does this, and the mitochondria does this. Okay, now there's a few that you kind of always end up doing that with. But really think about it as, what if I wanted to make a protein? What would I do? And then take yourself on a little road trip. And it's a lot more interesting and a lot less memorizing. Okay, so let's start at the very center, which is also the brain, so to speak, of the cell, the nucleus. Okay. So the nucleus is sort of the brain, okay, or I like to keep referring to it as the library of the cell. Okay, so this has all of the information for everything that happens in the cell, and as such ends up sort of controlling everything that happens in the cell, right? Because you have to come here to get that information. Okay. All of this information is wrapped up in that DNA. Okay, so DNA is the biggest word. So when we say DNA, this is everything. Okay, the biggest umbrella word. Okay. We're also going to use these words chromosomes a lot. Okay, a chromosome is basically a subunit of DNA. Okay, so if we took that DNA and parsed it out into little boxes or little pieces, okay, I have one large DNA, and I can break that up into 26 chromosomes, or 26 different pieces. Okay, you have one textbook broken up into 26 chapters, right? So that's a chromosome. That make sense? Okay, so we're still talking about the same thing. It's the same information. It's basically just saying, are we talking about all of it at once, or are we just talking about one specific part of it at a time? Okay, so remember, this DNA or these chromosomes contain all of the information of the cell. Okay, all of the information for how to grow, all of the information for how to reproduce, all of the information for the cell that you're in, if I'm a liver cell, but it also contains all the information for everything else too, for skin cells, for eye cells, okay, for nails, whatever it is, all of that information is there. Remember, it's not all being used, okay, but it all has to be there, right? Because we never want to lose any information. Information is precious. Okay. So the DNA is found inside the nucleus, okay, which is encased in the nuclear envelope. So the envelope is like the walls of the library. And this is what keeps everything safe. This is something that really keeps eukaryotes different from prokaryotes. Okay. So we're going to keep our DNA separate from even our cytoplasm. Because you never know what even gets into the cytoplasm. We've got food chunks floating around in there. We've got cell poo floating around in there. Okay. We can't risk the most important information in all celldom touching that stuff. Ew. Okay. Realistically, though, and very seriously, we can't honestly risk that stuff being damaged. Okay, because if you damage that information, you could damage the actual life of the cell, okay, or its future offspring from being able to access and utilize this information properly. Okay, so the walls of the library, okay, the nuclear envelope, ensure, kind of whistled there, ensure that this information remains safe and protected from everything that comes in and out of the cell at all times. Okay? So our DNA, our chromosomes, remain isolated from the rest of the cell at all times. Okay? And this is why that RNA that we talked about in biomolecules was so important, because only that stuff then leaves. Okay, so we copy information and it leaves the nucleus, but nothing goes in. Right? So when you go into a library, you copy information and you leave, but you're not ever going to take core material out. Okay, so it's like one of those fancy restricted sections where the books are like chained to the wall. The books don't leave. DNA never leaves. Okay, this feel good? All right, so our nucleus is in a safe house. So let's look at how we might actually make something then. So if I have, let's see, DNA, that was in the nucleus, right? Let's put this somewhere you can read it. Okay. So then we just said in order to leave the nucleus, that you're going to have to copy it as RNA. Okay, that was like you copying information. That was our big story from biomolecules. Okay, so this is what we learned in biomolecules. DNA stores information, RNA uses information. Okay, but uses it for what? That was sort of where we left that story dead, right? So what? Uses it for what? Okay, let's find out. So if RNA leaves the nucleus, the where is where we're going to go. Okay, so where we're going to go next is the ribosome. And that's where our RNA actually takes the information that it copied. I'll actually use here. Okay. So we'll talk about what the RNA does with the ribosome on Monday. So if you didn't copy this, this is fine. This is where we're going to pick up. Meow. And find out what exactly is RNA going to do. All right, don't forget to do your Socrative, last name, first name. My teacher's cue will be that we left on the ribosome slide, just barely, right? Can you stop recording for me, Jasmine?